So today I'll be speaking about our task where we are trying to design inhibitors for cancer therapy. In this, we are doing in silico strategies for designing and synthesis of novel inhibitors for cancer therapy. So when we talk about drug discovery is nothing but earlier it was like a serendipity where it was done um, uh, randomly by conducting some experiments without any theory to proceed for some kind of drug molecules. But it was like when we talk about drug research, it is like a needle, searching a needle in a haystack. And when we talk about modern drug discovery process, we can say we have certain steps which we have to follow for the drug discovery. And in the initial step, we try to understand what are the targets which we have to identify so that that target which is causing the disease has to be identified first. And then we have to validate whether that is the target which is causing the disease or not. And then go for small molecules which can inhibit this target. There we do a lead identification and then we try to optimize these leads so that we get some kind of structures which will be having maximum interactions with the receptor. That is the process which we do in lead identification and lead optimization. And once we get good molecules which are having better interactions, then we go for preclinical phase and then the drug discovery happens. The whole scenario takes approximately 10 to 15 years for a development of a typical drug molecule. And the research and development cost, workforce involved in it, clinical testing are quite expensive. And here comes computer aided drug design, where this target identification, target validation, lead identification, and lead optimization, these all can be done in computer. So there we talk about the computer aided drug design. What are the strategies which we have to follow? Do we have a validated target? Do we have active ligands? Do we have both? It all depends upon these three questions which we ask when we talk about computer aided drug design. So when we are talking about computer aided drug designing, so we first of all try to understand whether we have a validated target. That is, do we have a protein already available and can we go for any kind of modifications in the structure? So the strategy here is we have to try to analyze what kind of molecules, proteins we have. Do we have any known protein structures? Do we have any known ligands? Based on that, we go for structure-based drug designing, which is nothing but protein ligand docking. And if suppose we don't have any protein structures, but we are trying to go for a known ligand, we have known ligands but no known structure available in this in the database, then we go for ligand based drug designing. That is, if we have one or more ligands, we go for similarity search. If we have several ligands, we go for pharmacophore searching. And if we have more than 20 molecules, we go for quantitative structure activity relationship. So these are the two strategies which are generally used when we talk about computer aided drug designing, that is structure based drug designing and ligand based drug design. There is another such strategy which is called as de novo drug design, which means that we are trying to see as to if there is known protein but there are no ligands available in the literature, then we go for de novo drug designing or virtual high throughput screening. That is, de novo means from the scratch. You are trying to design your own molecule from the beginning and trying to analyze whether this molecule will be better fitting into the protein structure, active site, and then we try to analyze that. And if suppose you don't have any protein structure and no ligands are available, then CAD cannot be used. But there are probabilities that you can do homology modeling and try to analyze as to how the protein structure looks like. Then there are a lot of strategies which we use. There is AI model also available for doing homology modeling, which helps in looking at thousands of proteins available in the literature and getting back to the structure of requirement. If you give the pasta sequence to the uh, AI, it tries to give you the structure accordingly. So that is the process which we generally do in homology modeling. And then we try to analyze what is the active site in the protein and then do a de novo design. First of all, once you design the protein, you have to check for the uh, authenticity of the protein, whether it is correct structure or not. For that, we have ProCheck, Ramchandran plot, which we call it as, and PROSA. All these we try to analyze to see whether we have homology model structure is correct or not. And finally, we try to get new molecules. Okay, so some problem not going. So, what is computer aided drug designing? When we talk about computer aided drug designing, we talk about Emil Fisher's lock and key mechanism, where we say the keyhole of the lock is like the active site of the protein, and we try to design a lot of molecules which can open the keyhole or which can fit into the keyhole. That is the strategy which was used earlier. But now what we do is we try to understand the architecture of the protein active site and design the molecules computationally, come up with 10 or 15 molecules or 20 molecules, and then try to see how they are interacting with the molecule, with the active site, and then try to synthesize those molecules. And this is the best strategy which we use for computer aided drug design. So what is docking? Docking, we say it is an attempt to have a best fit between two molecules, a larger molecule and a smaller molecule, that is a receptor and a ligand. Or you can say a protein and a small molecule, which we can talk about. So when we talk about docking, there are how the molecule exactly docks into the structure, that is a ligand. If you have a receptor, how the ligand is going and docking into this, and according to how it is fitting into the molecule, we can score the, and this is called a scoring. So best score will be when it is having maximum interactions with active site. Not only that, we have to also see how the molecule is going into the active site, having different poses at the active site in the binding site, and then gives the scoring function. So these two, which is pose and score, are very, very important when we talk about docking methodologies. And this scoring function can be a global minima or a local minima. When we say it is not fitting properly into the active site, then we say it is a local minima. And when it is fitting properly into the active site, it gives a minimum energy, which is called as a global minima. So these are some of the strategies which we use. Now, this is one of the binding site of the protein, and ligand comes, checks the binding site, has different poses around this active site, and the one which is best fitting into that will be taken into consideration. And there are other strategies which we come up of, like complementarity, cooperativity, chelate effect, pre-organization, molecular fit. What is complementarity is how best the receptor and the ligand are fitting properly is what is called as complementarity. Cooperativity is like we are trying to see whether the ligand is going and fitting into the active site or the allosteric site of the protein. And if there is a possibility, then we say there is a cooperation between the allosteric site and the active site, and based on that, they will fit properly into the active site of the protein. There is another strategy called as chelating effect. Maximum interactions when the ligand has with the receptor, then we call it as a chelating effect. More number of interactions, best is the binding with the receptor. The other one is pre-organization, where we say that the active site of the protein is already pre-organized so that the ligand which is coming and fitting into the active site has to check where it has to exactly go, which pose will have the better binding affinity is what we try to see in the pre-organization. The other one is called as molecular cleft, where the cavity or the active site is deep inside the protein, and when you have such kind of protein, how the ligand has to be designed so that it goes into that cleft and has maximum interactions, non-covalent bond interactions like pi-pi stacking and electrostatic interactions. So these are some of the non-covalent bond interactions which we can talk about. So maximum the non-covalent bond interactions, better will be the binding between the receptor and the protein ligand. So these are some of the non-covalent bond interactions which we talk about when we talk about these uh, non-covalent bond interactions. One is ion-ion interaction. You can see the strengths in kilojoules per mole, and ion dipole moment interaction, dipole-dipole interaction, hydrogen bonding, cation pi interactions, van der Waals, and hydrophobic interaction. These are some of the non-covalent bond interactions which we try to see when we are talking about protein ligand interaction. And this is a fundamental binding affinity or the equation which we take into consideration, where we say receptor plus ligand will give you an association constant by dissociation constant and the complex. So if you want to calculate the association or the dissociation, then this is the equation which we use. And depending upon how you are doing, minus RT ln Ka is what the free energy calculation equation which we get. With the free energy, what we have, we can calculate the enthalpy and entropy of the interaction and then say that which one is better having a binding affinity with the receptor. So these are some of the docking programs which we generally use: FlexX, Gold, Autodock, Glide, induced speed docking, and Dock 3.6. We also use, we generally use Glide as the software for docking protocols. Sometimes we also use Autodock, but Glide is a paid version and Autodock is a free software. So if you are interested, you can go for Autodock. FlexX is there in Sybil software. That is also a paid software. So I will just go into my work, what we have done and how we have gone for the in silico strategies for synthesizing and evaluating the quinoline derivatives which have targeted VGFR2. So we have gone for VGFR receptor, where it is actually uh, affects the signaling pathway, which is PI3K AKT mTOR pathway, which is help dysregulation of this causes the cancer. So we say that EGFR, VGFR are converging to PI3K AKT mTOR pathway, which causes angiogenesis and proliferation. Based on this, we went for literature search, where we have taken all the VGFR inhibitors which are available, which are used for inhibiting the vascular endothelial growth factor receptor, which actually stops the activation pathway, which leads to the cancer. So based on this strategy, we have taken more than 100 molecules to see how they align when we take only the ligands into consideration, which is nothing but pharmacophore search. So we have aligned active and inactive molecules to see as to how they align. And when we align the active molecules, we got the pharmacophore as acceptor, donor, hydrophobic, and two rings are very, very important for this kind of structure to be having better interactions with the active site. And also we have done a scatter plot where we have taken a experimental PIC50 and predicted PIC50 and tried to see how they align. And we got a good alignment where we got a regression coefficient of 0.9, which is very, very good. So we went on for looking at the quantitative structure activity relationship visualization, where we try to see what is the pharmacophoric features, what are the hydrophobic features which we have, and what are the uh, combined effects which we call as electronegative potentials and electropositive potentials which are present in this molecule. And also when we dock this molecule, we have seen that this interaction which we are calling as 16919 is very, very important interaction which has to be there if you want the molecule to be having a better binding affinity. So based on these strategies, we went for the structural requirements. First one is we should have a acceptor, donor, hydrophobic, and two rings which have to be there in the structure. And there should be a hydrogen bond interaction with cysteine 919. And also the aromatic ring should protrude into the hydrophobic cavity, which is present in the DGF model. Based on that, we have taken all the molecules which are there in the literature, drug molecules like vanitamib, cabazanib, and pazopanib. And we try to analyze what are the pharmacophoric features which are present here and try to design our own hybrid molecule for VGFR2 as VGFR2 inhibitor. So this had a quinoline moiety and this had a dimethoxyphenyl group and this had 4-anilinoquinoline moiety, this moiety, and this had 2-aminopyridine moiety. So all these were taken into consideration to make the hybrid molecule. So that we got 2-amino 4-anilino 6,7 dimethoxyquinoline derivative. So based on this, we have seen whether we have this pharmacophoric features which are required for this molecule. And we came up with an acceptor, a donor, a hydrophobic group, and two rings were also present in this structure. So we designed some more molecules to understand how these will be and try to synthesize these molecules using this strategy where we have taken methyl 4,5-dimethoxyanthrylene and try to cyclize this molecule. And then we got this molecule, the final molecule. And then we went for checking the binding pose of sunitinib, already existing drug molecule, where this molecule was not having the interaction, which was very, very important, that is cysteine 919. So only it had some other interaction. So when we checked our newly designed molecule, this interaction was there. And also it was fitting properly into the active site of the protein. So this is the hypothesis which we talked about. And it was having the same hypothesis of acceptor, donor, hydrophobic, and two ring system. And also the alignment was properly in matching with the alignment of the already existing ligands. Now, this is the glide pose of the molecule where you can see the interactions properly here, and the dock score was minus 10.76 kilocals per mole, and it had cysteine 919 interaction, which is a crucial interaction, and also other interactions. So we went for checking the binding inhibition with the VGFR2 in comparison with sunitinib. Sunitinib had a binding affinity of 19.1 micromolar, whereas our molecules PIH and PIB had 17.1 and 17.5 micromolar. So based on this, we thought these two molecules are having better inhibitory activity compared to sunitinib, So we went for other cell viability assays like HeLa cell, uh, HeLa and cervical cancer cells. And we could see that these molecules were having 52.9 micromolar, 16.8 and 10.7 micromolar at 50 micromolar concentration. So these three molecules were having good uh, expressed good cell viability accordingly. So we went for chick embryo angiogenesis, where we have taken 0 hours and 4 hours, where we took untreated DMSO, 5B and 5H and sunitinib molecule. And we have seen that the angiogenesis has reduced considerably from 0 to 4 hours in both the cases. And also in sunitinib, we could see. So we can say that accordingly that 5H and 5B, which we have strategically uh, designed using computational methods and then synthesized and got these molecules, had better anti-cancer activity. So this is one of the strategy which we have used, where we have taken 3D quadrilateral analysis, and then we took all the FDA approved drugs, tried to design our own molecule based on the already existing drug molecules, and then we went for docking, and then we checked the binding inhibition with VGFR2, in vitro chick embryo angiogenesis, and cell viability assays. And we could say that 5H and 5B, these are two molecules which have come up as best inhibitors for VGFR2. And then we also took the PI3K AKT mTOR signaling pathway to understand how the AKT can be targeted so that we can have better inhibitors for the cancer treatment. So this is the signaling pathway for PI3K AKT mTOR, where PI3K has, which is phosphorylated, converts the phosphoinoside diphosphate into phosphoinoside triphosphate. And with that, AKT gets phosphorylated. Then the whole process takes place where it helps in cell proliferation, cell cycle progression, and all the processes, survival, growth, cell survival, growth, proliferation, metabolism, angiogenesis, all these happen in this cascade of signaling, which is PI3K AKT mTOR pathway. And AKT is nothing but a threonine, serine threonine kinase. So we wanted to target this, and we went for all the known PI3K AKT inhibitors which are there in the literature, and we did a literature review to see any indole derivatives are present which are having this PI3K AKT inhibition. And accordingly, we have gone for all these molecules were available in the literature which had indole moiety in them and having anti-cancer activity. So based on this, we went for the strategic design of these molecules, where we took AD412, which is in pipeline as a drug molecule, and try to structurally modify this molecule by replacing or by adding fluorine at this phenyl ring of the indole, so that there will be a lipophilic nature, and also fluorines always have some kind of good uh, as drug molecules. And also, not only that, the strategy was we have removed this aromatic ring, and there is only hydrogen present here. And at this juncture, we tried to put an R group where we could do structural activity relationship, which we can explore. And also, we put a biphenyl ring instead of pyridine ring to understand how this ring will enhance the activity. So these were the strategies for designing the new indole analogs, and we went for synthesizing these molecules, where we have synthesized 12 molecules with a simple strategy of synthesizing, where we took 5-fluoroindole and reacted with acrylic acid in presence of acetic acid to get the substituted acid group on the third position of the indole, and then try to um, uh, couple this with condense the acid and amine so that we get the group. And then we went for the Chonagashira coupling, where we got the biphenyl group here. And the amine was synthesized by reducing the nitro group to NH2. And also, not only that, where we have only one fluorine, we also try to check if we can have three fluorine atoms on the aromatic ring, how this will be affecting. And another 12 molecules were synthesized using trifluoro substituted aromatic bromo substituted amine, and then try to club it with the indole group to get this moiety. And when we checked for the cervical cancer, like HeLa and SIHA, we could see that apart from the buparlisib moiety, which was having 11.87 and 9.6, our molecule T5 was having 9.366 and 8.475 micromolar um, concentration it was inhibiting. So we thought these molecules are good. And also we checked for the normal cell activity, and we could see that this was having 51.75 inhibition, which is not so good. We are trying to get this into a better fitting thing, that is in process. And we also checked for cell apoptosis, and then wound healing scratching assay and Western blot. And all these have told that T5 is the best molecule amongst all the things. And we did the docking, where we took the indole moiety derivatives and checked how they are fitting into the active site of the protein, which is 2WXP. And we could see that the co-crystal ligand, which is in yellow ball and stick, and our molecules, which are in green. And also, we have AD412, which we have taken as a standard, also was docked, which is shown in blue ball and stick. So all these show that these molecules, which are 5-fluoroindole derivatives, were fitting properly into the active site. And also docking pictures are shown here, which show better binding interactions with the active site of the protein. And this was published in Journal of Molecular Structure. And I'll just quickly go through one more, where we have taken indole 134 thiodiazole shifts bases as anti-cancer agents. And here also we have looked for indole derivatives, how these, when clubbed with 134 thiodiazole scaffold, will they be acting as drug molecules? So we took the indibulin and acetazolinamide and hybridized this molecule to get a indole 134 thiodiazole hybrid. And we try to synthesize this molecule shifts base by clubbing the aldehyde with the amine to get the shift space. And we have gone for the in vitro anti-cancer activity of these molecules, and we could see that these molecules T43 and T48 had better binding affinity, had better anti-cancer activity in vitro compared to other molecules. And these are some of the docking pictures of these structures, where you can see the indole 134 thiodiazole hybrid is are as yellow ball and stick model, and co-crystal ligand is there in blue. And we could see that the alignment of these molecules are better in the active site compared to the other. And when we check the ligand interaction diagram of the existing drug molecule and our molecules, we could see there are maximum interactions which are happening with the active site amino acids compared to the existing drug molecule. And this we have published in ChemMedChem, and all these publications have acknowledged PAIR. Thank you. If you have any questions, you can please ask me. If there are no questions, I'll stop sharing so that the next person can start with the presentation. I think Sandhya is next. I'll just introduce Sandhya and then we can go for the.
Hello,
Hello?
I'm audible.
Yes, yes.
Yeah. Good morning, everyone. I'm really glad to be participating in this Osmania University Education Week. So, I'm here to present my webinar on integrated genomics for breast cancer. I work at Department of Genetics, University College of Science, Osmania University. So this is a part of the ANRF-PAIR project, which is entitled Integrated Multi-Omics Approach to Identify the Markers and Factors Driving Towards Drug Resistance in Triple Negative and Hereditary Breast Cancer. So I've been collaborating with Dr. Raja Mohan Roy, who is an associate professor at Department of Animal Biology, University of Hyderabad, for this work. And to begin with, we always have known that cancer is a challenging condition with growing health burden in India and globally too. So, I would like to give a little bit of context about cancers and the molecular complexity. There are around more than 120 different types of cancers, which are classified based on the cell type in which the cancer originates, and based on the histology, and based on whether it is malignant or benign. And there are diverse molecular alterations that are involved from origin to progression. So basically, the altered cell behavior triggers the cell transformation from a normal to a malignant form. And this malignant transformation proceeds, and the cell proliferates abnormally, and it can lead to distant metastasis and spreading of cancer from one place to different sites in human. Molecularly, there are different genes and mutations which are implicated in cancers. Basically, they could be oncogenes or tumor suppressor genes, or genes that regulate apoptosis or DNA repair genes. And most of these mutations are acquired, and very few genes or mutations are hereditary in different types of cancers. Most importantly, cancer genomes are dynamic. It is not a single disease, and the genome changes. It's not a static thing. So, tumor evolves constantly, continuously over time, and it accumulates different types of mutations. And tumor heterogeneity, like intratumor and intertumor heterogeneity, is one of the biggest challenges right now in cancers. Uh, Comparing the normal cells with that of the cancer cells, there are distinct features, and the morphological and also at the functional level. So there are several molecular properties that underlie cancer, which are referred as hallmarks of cancer. It's namely something like avoiding the immune destruction. The cancer cells have enormous replicative potential. Then also it evades the immune system, and with many more. Coming to the today's topic, that is breast cancer. Breast cancer is a type of cancer that originates in the breast in different parts of the cells, like lobules or ducts or nodes. In our country, around 1 in 8 women are diagnosed to be having cancers, and every 4 minutes, one woman is diagnosed of this breast cancer, and every 13 minutes, a woman can succumb to this breast cancer. Histologically, there are different types, as I told, lobular, ductular, and also based on its invasive nature, they're also named as invasive and non-invasive. And they are graded based on the TNM system of staging, that is based on the tumor size, based on the involvement of lymph node, and whether it is metastatable or not. And molecularly, also, the breast cancer can be classified as luminal A, luminal B, HER2, and triple negative. And coming to the genetic aspects, as I told you, only 5 to 10% of breast cancers are hereditary, and majorly it is acquired with involvement of mutations in high penetrant and low penetrant mutations. Majorly, BRCA1, BRCA2 mutations are implicated in breast cancer. Uh, and there are several risk factors identified for breast cancers. And initially, based on the symptoms, it can be diagnosed clinically with mammography, ultrasonography, or magnetic resonance imaging and all. But the breast cancer susceptibility can also be predicted based on the implication of different risk factors that are implicated. Coming to the statistics, breast cancer ranks first in incidence among all other cancers, accounting to 11.7% of cancers, and fifth in terms of mortality, as per the recent statistics. And and 46.6% of cases are reported mostly in 30 to 40 age group. Although predominant in this age group, the onset of this condition is increasing in younger cases, particularly in India and Telangana. And most of half of these cases which are reported are in stage 3 and stage 4. 70% of the cases in advanced stage have poor survival and high mortality. And in India, around 14% of cancers account to breast cancer and leading to 50% of mortality. Mostly it could be because of lack of awareness, early screening, and late diagnosis, which are major reasons for the low breast cancer survival in India. Comparing the global statistics with that of Indian, as I said, the major cancers, out of major cancers, breast cancer incidence has increased alarmingly in 2025 compared to 2015 statistics. And the incidence are also very, very high in the younger onset cases, and particularly in Telangana, on an average, 31.3% of the cases are breast cancer cases, and 40.2 are percent of incidences in Telangana, which is very high. And triple negative breast cancer is one heterogeneous subtype of breast cancer comprising 12 to 17% of all breast cancers. And around 5 to 20% account for germline mutations, that is BRCA1 or BRCA2. And 70% of them are arising in BRCA1 and 23% in BRCA2. And the incidence varies from country to country, from 6.7 to 27.9. And in India, it is reported very high. And TNBC tends to behave more aggressively compared to other subtypes because of lack of the three important receptors, that is estrogen receptor, progesterone receptor, and HER2 receptor. Because of lack of these three receptors, they are having a limited options, therapeutic options. And among the TNBC, there are several molecular subtypes, majorly 6 subtypes. And the treatment, since the treatment options are limited, the 5-year survival rate is also very, very low. That is from 37 to 77%. Conventionally, breast cancer therapy, therapies have been very, very prominent in India. But the advanced therapies involve, or the targeted therapies are very, very. It's very limited in some of the countries. So emerging therapies include the use of HDAC inhibitors or immunotherapies or nanotherapeutics. But from research point of view, these cancers are very difficult to be targeted. Hence, the studies have focused majorly on identification of the actionable drug targets in TNBC. And particularly in view of the recurrent cancers, because around 30% of TNBC cancers have a high recurrence rate, and it is incurable and is lethal. So chemotherapeutic resistance, what are the genes involved, what are the molecular mechanisms involved, is a major focus of the research in recent days. So to sum up, the clinical challenges are the delayed diagnosis, heterogeneous molecular profiles in different subtypes of cancer, particularly in subtypes of TNBC. And people, because of this molecular diversity, they show differential drug responses. And there are lack of options for these aggressive cancers. Therapeutic failure is major because of primary or secondary drug resistance, because of increased genomic instability. And also side effects are are high or many due to chemotherapy. So currently there are no specific markers for risk prediction or for diagnosis of this subtypes of cancer, and metastasis prediction is far away, and therapeutic monitoring is very, very challenging. So the context is that the genome analysis can be an important tool for clinical at a clinical level, particularly to identify the tumor type at the beginning of at the time of diagnosis, recognize if there are any hereditary mutations implicated, and identify the appropriate therapeutic targets, determine the prognostic risk score for each individual, and identify the markers for prediction for monitoring the treatment response. So in this direction, I can give you a couple of examples where most important one is there are several mutations that happen from the origin to progression in different individuals. So identification of driver mutations is very, very important, and of which of these are actionable. So one strategy is, first of all, list out all the mutations and classify or prioritize the driver mutations amongst these, and see which of them are appropriate for designing some or can be which of them these targets can be used for as a therapeutic targets. Second important application would be because we all know that a single gene may be associated with multiple cancers, and a prominent one like TP53, or multiple genes can increase the risk of a single type of cancer. Like in breast cancer, there are BRCA1, BRCA2 majorly, and other high penetrant mutations like PTEN, ATM, PAL2, CHEK2, RAD51, and so on. So what are although we have identified several mutations in these low penetrant and high penetrant genes, how can they be useful for characterization of the tumor in the beginning, and so that appropriate therapies can be planned? That is the concern. Coming to the molecular subclassification of this tumors at diagnosis, as I told, the mostly clinically only the patient has been are being stratified, but molecularly if we can stratify at diagnosis, it will be really, really very helpful for from clinical point of view, and the patient would benefit from the available therapies. So third important thing is predictive and preventive testing. So in our study, we aimed to study some of the hereditary mutations because for a long time we have been focusing only on BRCA1, BRCA2, but other hereditary mutations, although rare, what are they, and how they could be contributing to breast cancer pathogens is an important area. And screening these germline variants apart from BRCA1, BRCA2 can help in counseling the other members and warning the other members of the family, particularly if there is an incidence of male breast cancer patient. So the female members of the family have to be warned, and they have to be screened as a preventive strategy. But further, there may be a chance of recurrence in the affected individuals. So the recurrent risk also can be done based on the molecular profiling. If you see in the breast cancer, 12.9% is accountable, and about 87% of risk, 87% risk can be predicted from her germline mutations. These are the different technologies. Mostly the NGS applications, in order to understand the genetic basis of all subtypes of breast cancer in a comprehensive manner, from genomics point of view, proteomics point of view, transcriptomic point of view, and epigenetic point of view. So these are the different clinical bioinformatics tools we are trying to apply for our experimental data in order to navigate through the prominent molecular mechanisms that are implicated in breast cancer, particularly TNBC and metastatic. So far, these are some of the genome profiling studies that have been conducted globally. And also some of them are recently in this decade in India, but there is a huge research gap. Substantial progress has been made, but there is a lack of integrated genomic and transcriptomic studies in India. Particularly, there is no clinical correlation between the experimental results, and because the validation also is far away, and the prognostic potential of the variants identified is also not being conducted. And most of the exome data was a focused analysis where they have included a set of genes or genes pertaining to a particular pathway, but a comprehensive exome data, whole exome data was not analyzed in India. And the sample size was low, and mostly the samples were also heterogeneous. Hence, based on these limitations, our study has been systematically designed to include all the clinical data, include enough number of cases for each subtype, and screen both for germline variants and as well as somatic variants, and with a focus to investigate the long forgotten regulators like RNA regulators, wherein we already know that long non-coding RNAs and mRNAs are one of the prominent partners in the different oncogenic regulatory networks of different cancers. So in TNBC subtype where limited therapeutic options are there, so our focus has been on understanding these nodal networks in the onset as well as progression of TNBC. Furthermore, most of this the variants have been analyzed from tissue point of view. Only tumor specific variants have been identified. But are there any variants that we can identify in a sample where the serum can be used, where minimally invasive biomarkers can be identified? That is one of the concerns. So the key objectives of the study has been start analyzing the genomic and serum transcriptomic analysis, particularly comparing the TNBC and non-TNBC to derive the potential panel specific for the TNBC in India, and identify relevant LNC-RNA, miRNA nodal networks with clinical utility, and a genomic screening of familial breast cancer samples by targeted exome sequencing to identify the non-genetic variants in the non-BRCA germline variants with clinical significance. The study is expected to identify the serum and tumor biomarkers associated with each breast cancer subtype and identify common specific mutations for TNBC, which can be used to suggest a panel for preventive and predictive screening in India, and define alterations in the key signaling pathways to understand the molecular significance of the variants with regard to drug resistance and design of novel therapeutic alternatives, and also understand the role of foreign regulators in drug resistant mechanisms. So this is a methodology we have adopted. There are two components for this study. One is comparing the TNBC versus the non-TNBC cohort study, where all cases of TNBC right from diagnosis to including the metastatic breast cancers will be taken up. And and these cases will be analyzed both at the serum level, at the using the samples, that is blood sample and the tissue samples. So the variants will be analyzed by subjecting them to whole exome sequencing as well as transcriptome profiling, and the data will be analyzed where by all the computational tools will be applied to identify the actionable driver variants, which and the differentially expressed genes will be validated on a large cohort later to see the prognostic significance. The second component is the hereditary breast cancer family studies, because there are very few family-based studies where mostly they were only focusing on BRCA2, BRCA1, BRCA2, as I said. So we are recruiting around 4 to 5 breast cancer families where linkage-based analysis will be conducted and targeted exome sequencing will be conducted to study the non-BRCA germline variants with clinical importance. So far, we have searched all the publicly available databases on TNBC and other subtypes, and these are the common global mutations that have been identified, mostly the TP53 with 60 to 80% frequency, then PIK3CA and other genes. So we have also screened some of these genes individually on a large cohort where we only found 6% of PIK3CA mutations. So this is the data which I'm showing, which has been conducted for the past 2 years in collaboration with the Genome Foundation and on 30 TNBC patients. So these are the tumor-specific TNBC mutations, particularly if you see PIK3CA1 was identified in major patients, but we could not identify that were reported globally. but we are trying to understand how they are implicated and what could be the other variants specifically arising in the tumor, TNBC tumors. And simultaneously, we have also screened for the long non-coding RNAs involved in the breast cancer. And from the literature, we have identified and prioritized the LNCs, particularly NB81, hot air, and Andrela. So these are the LNC RNAs which we are now validating on the in the serum as well as the tissue level. So these are the serum level LNC RNAs in the breast cancer. There are so many LNCs that are upregulated and can be used as a diagnostic markers, particularly from metastatic point of view. Also, we have identified a couple of serum long encoding RNAs, of which we are planning to analyze H19, Nandrel, and hot air for now in this study. So also we have looked at the serum microRNAs in the breast cancer, which are differentially regulated, differentially expressed in the breast cancer at different stages, both at diagnosis as well as during therapy, or also in resistant cases. So combining all this high prioritized LNC RNAs, miRNAs, and the variants, we are trying to generate nodal networks and the significant pathways that can be targeted with available molecular targeted therapies. So, in summary, comprehensive studies are very integrating targeted exome sequencing, serum transcriptomics, and system biology approaches are very, very essential to identify the TNBC specific biomarker panels and characterize the clinically significant non-BRCA germline variants too, understand the regulatory RNA interaction networks, and improve the early diagnosis and prognostic prediction, enable the personalized therapeutic strategies in Indian breast cancer patients. Thank you. So far, we have collected around 6 TNBC patients, recruited TN6 PNP patients further only for the study. And the experimental data has to be analyzed. So we are just waiting for the results. Thank you.
If you have any questions, please you can ask. If there are no questions, then we'll invite Professor H. Surekarani, ma'am, to talk. Her title is Precision Oncology Drug Targeting Breast Cancer Mutations, and her hub co-PI is Professor M. Brahmanandam. So I invite you, ma'am, to deliver your talk.
Good morning, ma'am. Ma'am, am I audible? Hello?
Yes, ma'am. Yes, ma'am, you are audible. Good morning.
Yeah. Good morning, ma'am. Good morning, ma'am. So, the project director, Professor Brahmananda Manamati from the University of Hyderabad, principal investigator, senior Professor M. Vijulata madam, and all the co-investigators for the ANRF-PAIR project titled Innovations in Health and Medical Technologies, Sustainable Health for the Resilient Future. In this, the work package 1 is for biomarker identification, and sub-package 1.1 is to identifying the biomarkers for biomics approaches. And I'm Professor H. Surekarani, Head Department of Genetics, Usmania University, and co-principal investigator for this project. And Professor Brahmananda Manavati is the investigator from the Hub University. And for the task 05, entitled Genetic Polymorphisms Associated in the HECT family ubiquitin ligase genes in breast cancer patients as potential biomarkers. So, Dr. Sandhya has just now discussed about the breast cancer in detail, about the pathology, the modalities of the treatment, and then the different types. But I will be reiterating some of the important points regarding the breast cancer. So, as you all know, this is the leading cause of cancer mortality in women, and it accounts for the 31% of the new diagnosed female cancers worldwide. And in India, the incidence has increased significantly. 13.5% of all the newly diagnosed cases are the breast cancers. So, in spite of these advanced technologies in medical field, still it continues to be the lethal malignancy due to the lack of early detection, screening strategies, and ineffective therapeutics for the late stage metastasis. The breast cancer, it has been staged using the TNM system, that is the tumor size, lymph node status, and then the metastasis. And invasive ductal carcinoma, this is the most common type of the breast cancer, which is representing the 80% of all the breast cancer diagnoses. And the patients are staged either as early stage or the late stage. So in the early stage, it shows that the tumor is more than 2 centimeter, but not more than the 5 centimeter. Where in the late stage, so it includes the T3 and T4, where the size of the tumor has been increased, that is, which is more than 5 centimeter, and it might be growing into the chest wall or the skin. And as you all know, there are many risk factors for the breast cancers, both which are modifiable and then the non-modifiable. The modifiable will be including the radiation exposure, hormone replacement therapy, alcohol, tobacco, and a diet. And the non-modifiable are the age, female gender, genetic factors, menstrual and reproductive factors, family history of the breast cancer, and then the history of previous breast cancers. And as you all know, that there are mutations in the BRCA1, BRCA2, and P53, as well as in DNA repair genes such as ATM, have shown the significant association with the breast cancer development. And then the genetic polymorphism. So as you all know, the breast cancer is a genetically and clinically heterogeneous disease. So it has both the genetic factors and then the environmental factors, which are the contributors. And SNPs are the most common type of genetic variations in the humans. These single nucleotide polymorphisms, as well as the pathogenic mutations in the genes, predispose the individuals to develop the cancer. So these SNPs have only moderate risk on their own, but when they are combined with the additional non-genetic risk variables, their polygenic risk score becomes crucial and helps to predict the etiology of the breast cancer. And these single nucleotide polymorphism may be present in different components of the gene. So they might be present either in the promoter region. So when they are present in the promoter region, so it will be affecting the transcription factor binding and then resulting in the differential expression of the genes. And these might be also present in the 5' UTR region, so where it might lead to the transcript stabilization of the mRNA. And even the SNPs can be present in the introns also. So when they are present, so it might be altering the splicing, and also the exon skipping might be occurring. And introns might also be present in the exon regions, so where it might be contributing to the amino acid replacement, so leading to the protein structure modification. And further, these SNPs may also be present in the UTR region. So similarly, so the modification of the transport stabilization and mRNA localization in the cytoplasm might be occurring. So these SNPs, so these are also again categorized into different types, like the synonymous SNPs or the silent SNPs, where there is no amino acid change and it might affect the splicing or the expression. And the non-synonymous SNP, whereas it is also called as a missense, where there is an amino acid change which potentially alters the protein function. And there are nonsense SNPs, so which might lead to the premature stop codon initiation, so which leads to the truncated or the non-functional protein. And then the splice site SNPs, so which will be disrupting the splice signals, leading to the aberrant mRNA processing. And these SNPs which are present in the promoters, these are which are called as the promoter SNPs, they might alter the transcription factor binding, so which might lead to the changes in the expression. And there are different methods for the detection of these genotyping methods, like the PCR RFLP, which is a restriction fragment length polymorphism, Tachman assay, that is allele-specific real-time PCR, SNP arrays, that is high throughput genotyping, microarrays, GWAS, that the genome-wide association studies, Sanger sequencing, which is a gold standard for the variant confirmation, and the next generation sequencing, which is very comprehensive for the SNP discovery. So this next generation sequencing has revolutionized the identification and analysis of the single nucleotide polymorphisms, allowing for a rapid high throughput detection of millions of variants across the genome. So in this next generation sequencing, so the DNA will be extracted and then followed by the library preparation and then the sequencing, followed by the reference followed by the reference assembling to the reference genome, and then the variant calling to identify the SNPs. So unlike the traditional methods, this NGS guided SNP discovery enables the precise genome-wide or targeted identification of the base level variations which are critical for the precision medicine. And this is the task 5. The objective of this present proposal is to investigate the genetic variants of the HECT family ubiquitin ligase genes in breast cancer as potential biomarkers for early diagnosis, prognosis, and for the targeted therapeutics. So as you all know, this ubiquitin protease system. This ubiquitin protease system is an important mechanism that regulates the protein levels in the body as well as the intracellular protein levels. It is used to degrade the misfolded, damaged, or the proteins which are not in use. So 80 to 90% of these proteins in the cells are degraded by this pathway. But now the question arises is, so how are these misfolded damaged proteins have been recognized? So for this, so there's a small protein which is called as a ubiquitin. So that functions as a molecular tag to mark the proteins for degradation or to participate in various cellular processes such as the DNA repair and protein localization. So this is attached to the proteins through a multi-step process, enzymatic process called the ubiquitination, involving 3 enzymes. That is the ubiquitination activating enzyme, ubiquitination conjugating enzyme, and in the ubiquitination ligases. So in the first step, the ubiquitination is activated by the UB activating enzyme, that is the E1, which is an ATP-driven process. And then this activated ubiquitin, so it has been conjugated to the to the ET, and this ET conjugated with the ubiquitin. Now the E3, which is the ubiquitin ligases, helps in targeting the ubiquitin to the target protein. That is a target protein. It might be a misfolded protein or the damaged protein. So the E3 helps in targeting the ubiquitin to this misfolded protein. So once the misfolded protein has been associated with this ubiquitin, so it forms a complex. So depending upon the ubiquitination, it might be a monoubiquitination, multi-ubiquitination, and a polyubiquitination. So once it is polyubiquitinated, then this protein, this target protein, is ready for the degradation in the proteasome. So the E3, the ligases, so it plays a very important role. That is, unlike E1 and E2, E3 ligases directly interact with the specific target proteins to ensure the correct substrate is modified. So E3 is very crucial. So in identifying the substrate that has to be degraded. So during this process, so it helps in regulating different cellular processes, like it controls the crucial cellular process including the apoptosis, signal transduction, and cell cycle progression. So it is involved in the different processes, and therefore it also acts as a disease drivers. That is, any dysregulation of E3 ligases, it is linked to many conditions including the cancer by influencing the oncogenes or the tumor suppressor. And interestingly, and these E3 ligases are also forming the therapeutic targets, because due to their precise control over the protein stability, E3s are significant targets for drug development, particularly in case of cancer. So these E3 ligases, so that they are classified into 3 families. That is a ring, really interesting new gene, which acts as a scaffold to facilitate the direct ubiquitin transfer from the E2 to the substrate. And then the HECT, that is a homologous to E6 associated protein carboxyl terminus, which forms a covalent intermediate with the ubiquitin before transferring it to the substrate. And the other family, it is a ring between the ring, that is also called as RBR. So it utilizes a hybrid mechanism that starts like a ring ligase and finishes like the HECT ligase. So among the 3, so we are interested in the HECT family, that is a homologous to E6 associated protein carboxyl terminus. And there are, again, it has been characterized that there are 28 human HECT E3 ligases which have been identified. And these are characterized by a 350 amino acid catalytic HECT domain with conserved catalytic cysteine. And these are again subdivided into 3 subfamilies. That is the NEDD4 subfamily, so which consists of 9 members, which is including the NEDD4, NEDD4L, its SUMRF1, SUMRF2, WWP1, etc. And then you have the another family as HERC subfamily, which consists of 6 members, HERC HERC1 to HERC6. And also there are 13 members in the other HECT group, so which constitutes the E6AP, HUV, E1, etc. And these HECT E3 ligases, so they are also involved in different signaling pathway regulation, like the Wnt signaling and then hippo signaling, hedgehog signaling, MEC signaling, NOT signaling. It's also involved in the TGF beta signaling and then PTN, PI3, AKT signaling and RAF, MEC and ERK signaling. So you can just see the different members of this family, so which are regulating different pathways. And these, as they are regulating different pathways, so it has also been found that they are also associated with the different diseases. So diseases relevant in the HEC3 ligases, so the cancers, so different types of cancers, it has been identified the different family members which are playing a key role, like in the breast cancer, E6AP, HES1, HER2, etc., cervical cancer, colorectal cancer, gastric cancer, lung cancer, prostate cancer, and then the Wilms tumors. And apart from that, it is also regulating several neurodegenerative disorders like the Alzheimer's disease and then the Huntington's disease, etc. And it has been found that it is also involved in the viral infections like the Ebola, hepatitis, HIV, HPV, influenza, etc. Apart from these, it is also implicated in the other diseases like the autism, osteoporosis, etc. But however, the comprehensive understanding of genetic variants in HECT family genes is less explored in Indian breast cancer patients. Therefore, the present study has been taken to identify the genetic variants, so which have been associated with the Indian breast cancer patients. So the methodology adopted for the present study is that in the present study, a total of 250 female study participants with 100 blood samples and 100 tissue samples, that is both 50 surrounding normal and 50 tumor tissue samples, will be collected from the MND Institute of Oncology and Regional Center, Hyderabad. And also the 100 blood samples from the healthy women who are free from any disease will be collected from the local population as healthy controls. So during the collection of the samples, so the data collection will also be done. So after the written informed consent form obtained from the breast cancer patients and the healthy controls, so detailed personal and then the medical history of the patients and the control subjects will be taken. So the selection criteria, so we have the both inclusion criteria and exclusion criteria. The inclusion criteria includes histopathologically confirmed primary BC cases by the medical oncologist at the MND Cancer Hospital, and the healthy controls who will be involved for this, they should be free of any previous malignancies or any other health conditions. And the exclusion criteria, so women with the chronic diseases and associated with any other cancers, so they will be excluded from this study. And all this for the collected samples. So we are thinking to use a next generation sequencing. So where the breast tumors and paired tissues from the affected Indian women will be collected from the MND Institute of Oncology, Hyderabad, after obtaining the IEC approvals. And then both the tumor and the paired normal tissues collected will be processed for isolating the DNA and then followed by the preparation of the library for detecting the SNPs and the genetic variants in the HECT ubiquitin ligase genes and paired end sequencing to generate the high coverage reads across the HECT family loci. And then further, the quality assessment will be done using the FastQC tool, and pre-processing like the adapter trimming and the removal of low quality reads using the trimemotic or the FastPA will be used. And the processed reads will be aligned with the human reference genome. And the aligned reads from the tumor and the paired tissues will be compared using the genome analysis toolkit to identify the tumor-specific somatic variants and to identify the germline polymorphisms, including the SNPs and the indels in the HECT family ubiquitin ligase genes. And further, to determine the functional significance and association with the breast cancer, annotate identified variants using tools like the ANOVA or the SNP-F. And then validation will be done using the Sanger sequencing. Further, the identified gene-identic polymorphism in the HET family ubiquitin ligase genes will be correlated with the therapy targets of the treatment regimens provided to patients to validate and to assess whether these HET ligase variants influence the therapeutic response and also the ubiquitin pathway alterations caused by the polymorphisms. And they will be also assess them for them as the potential for a predictive or the prognosis for the breast cancer. And further, the validation will be carried out in the large sample size by the PCR-based genotyping. So for the significance of the study, so the new diagnostic markers are needed to identify the individuals who are at highest risk for developing the breast cancer. So this study might help in identifying the biomarkers for the diagnosis of the individuals at risk of developing the breast cancer. So the identification, the study might also help in the identification of the prognostic markers, which might enable the oncologist to begin the tailoring treatment strategies to the individual patients. So the study has not yet been started. So the ethical clearance has been obtained from the MNG hospital. So we are due for the collection of the samples from the MNG hospital. So Professor Brahmananda Manavati sir has got the MOU done with the MNG hospital. So maybe after the summer, we have to start the collection of the samples and then initiate the process. So these are some of the references which have been used for this study. And then we highly thank the ANRF-PAIR for funding this project. And then thank you. So I think I have completed a little bit early, but if there are any questions, I'm ready to answer.
So if there are no questions, then we'll go for the next speaker. That is Dr. Someshwar Polla and Professor Manjula Banuri and Dr. D. Suman.
Ma'am, good morning, ma'am. I will be starting first. Shall I start first? I think
Yeah,
Dr.
sure,
Polla
sure.
can continue.
Sure, ma'am.
Yeah, I'm sharing my slides, ma'am.
Yeah. Sureka, ma'am.
Hello, ma'am.
Stop sharing, ma'am. You please
Yeah,
stop sharing.
Yeah. Tennis.
Yeah, we can see your PPT.
Okay, ma'am.
But put it in the presentation mode, ma'am.
Did it.
Now it is fine.
Yes.
You can
So, good morning, everyone. The title of this webinar presentation is Task 14, Subpackage 2.3 of ANRF-PAIR OU. So, this is femtosecond laser fabricated SIRS substrates integrated with lab-on-chip devices for non-invasive diagnostics of cancers. So, this is Professor Manjula Banuri, Department of Biochemistry, who is presenting, and this is about Task 14. So this, we have chosen endometriosis. So our lab has been working in this particular area for almost 25 years. So we have chosen this. So just brief, I would like to brief you with the disease. What is endometriosis? Endometriosis is a very common gynecological disorder. About 90 million women in the world will suffer with this, and the prevalence is 10 to 20% of the reproductive age women, and it is more common than breast cancer and cervical cancer. So what is it basically? So endometrium is the inner lining of the uterus where the implantation occurs if the fertilization takes place. But this inner lining will grow outside the uterus. If it is inside the uterus, it is called utopic. And when it grows outside the uterus, it is called ectopic. So ectopic growth of endometrium is considered as endometriosis. So it is highly estrogen and progesterone dependent disease. And then, so these are the different forms of endometriosis. It can manifest as adhesions. Two organs may be involved where adhesion will be there between two organs. And it can be a chocolate cyst on the ovary, or it can be a lesion. So it can, the most probable places of the manifestation of the disease is the pelvic region, and even the bowel, bladder, all others can also be involved. And the rare cases will be there in the other regions of the body. So the major aspect of this particular disease is debilitating pain. It's very painful disease, and pain during menstruation will be unbearable. So it is inherited because there is a risk in the, if one sister is inherited in the family, other sister is also having a risk of developing the disease. So it is known to be more prevalent in monozygotic twins, and also affected sister pairs are more likely to have severe disease. And it has even ovarian cancer link. It is basically not a cancer, but if it is present in the body, because there will be a lot of inflammation, and the chronic inflammation is not good for the body. So it may cause cancer, but the risk is a little low, but it can cause cancer, specifically ovarian cancer. So the pathogenesis, as of now, this is an enigmatic disease. It has many factors make the disease to happen. Several genes are involved. As of now, there is no non-invasive marker, and the diagnosis itself is a very, very complex and then challenging thing. So it is also termed as enigmatic disease because no proper known cause is documented. So the most probable patients will be suffering with infertility. So we get almost all enrolled patients will be infertile, because in India, the menstrual pain is considered as a very normal thing, and female will suffer and then they will take it. But once the fertility issues arise, then they will go to the hospitals, and most of our 90% of our enrolled cases are with infertility. So I told you there is a cancer risk too. So the diagnosis is a big challenge, I was telling you. So the gold standard is laparoscopy, which is invasive, and then biopsy is preferable over visual inspection. We can even diagnose with, to a certain extent, with pelvic examination and imaging techniques. But as of now, there is no non-invasive marker, and the disease has overlapping symptoms with several pelvic disorders, even with IBS also. So it is very difficult to diagnose the disease, and a female will suffer for 10 to 20 years, irrespective of any region or any country in the world. 10 to 20 years it will take to diagnose. So their lives, if the diagnosis is not there, treatment will be very difficult. So this is the laparoscopy. This is an image of laparoscopy. There you can see in the shot, you can see the lesions of the endometriosis. So treatment options are surgical and medical, and then surgical involves conservative and non-conservative. So laparotomy is the first option, like laparoscopy plus the treatment of the endometriosis is laparotomy, where the lesions will be removed using laparoscopy. And then the pain management, because I told you it's a very, very painful disease, and hormones and painkillers are the major options. So just I will brief up what is the status of endometriosis now. There is no cure as such. Understanding the disease is very, very complex. As we are trying to understand the disease, it is becoming much more complex. So, and then and medical therapies and targets only the symptoms, not the root cause, and the side effects of the drugs are very, very bad. And even the surgery won't guarantee sometimes, and there is only at least 6% of the chances of the recurrence, even after the surgery is done, radical surgery, where the removal of the uterus, fallopian tubes, and ovaries are, even though they are removed, the recurrence is possible. So So our lab, I told you, we are working from past 25 years. From 2001, we are involved in identification of markers and other things. So I'll just brief up what we worked as of now, and then what we will be going to work with ANRF-PAIR. So we worked on mitochondrial. So we were working with any particular gene location is involved. Any particular gene or any other gene location, a panel of genes are involved in the disease, so that we can get a diagnostic marker and so that we can have a better therapy. When we work with whole mitochondrial genome, we are the first people in the world as such to document this, and we worked on copy number and D-loop, and we worked on nuclear genome like immunological factors and also tumor suppressor genes, even though it is not cancer, because any growth requires proliferation, and then tumor suppressor genes are involved in any growth in the body, even if it is ectopic growth of the endometrium. So, and we also worked with epigenetic factors, and we have developed a rat model of endometriosis. So actually endometriosis occurs only in the animals or only humans where the mammals, I mean to tell, where the menstrual cycle happens. So generally big cats and then rodents like rats and all, they don't have menstrual cycle. There is no shedding of the endometrium. So they have estrous cycle. So they don't have, so the spontaneous occurrence in rodents won't be there. So I would like to make a note that bats do have endometriosis. Bats are mammals. They do have a menstrual cycle, and even primates have a menstrual cycle. So the spontaneous occurrence of the disease happens only in the mammals where there is menstrual cycle. So we developed a rat model by surgically inducing. I told you the rats don't have a menstrual cycle, so we need to induce the disease surgically. So this we have developed, and then we started working with a compound called Sepia ink. Sepia is a cuttlefish which has an ink gland, and this particular ink is used in alternative medicines like homeo, but we have used whole ink, and then we wanted to decipher what is its role in endometriosis. Still the work is going on. It is with the DBT builder funding. And right now, coming to our project now, so we wanted to decipher what is the role of microRNAs in the endometriosis pathophysiology so that they can act as biomarkers. So, 2024 Nobel Prize goes to microRNA. So it is picking up, it is in the limelight more. So microRNA can modulate the gene expression. So it plays a very important role. There are at least 5,000 microRNAs in human beings. So from that, we need to decode which are playing a very important role. And even we wanted to check because the disease has overlapping symptoms with other IBS and then other pelvic disorders. So our microRNA panel should be able to even decode the differential diagnosis of the different diseases also. So that is the theme. Those are the objectives what we have framed. And the work what we started is from January 2026 and then till today. So I'll just brief up what all we have done. So the approved objectives are to develop a transformative diagnostic platform by integrating femtosecond laser fabricated surface enhanced Raman scattering, SIRS, substrates with the lab on chip. So we will be identifying from our lab, we'll identify the microRNA, and chemistry department, Dr. Pola, Someshwar Pola will code the microRNA with nanomaterial, and then this particular thing will be giving, we will be giving it to Hub, Dr. Ram Gopal, there from the physics department. So he will develop a spectrum where it can, even the very, very minute levels of microRNA can be detected, because our target is non-invasive identification of non-invasive biomarkers. So we are targeting either in the urine or in the saliva or in the blood. We will see how it progresses. So first we need to identify what is which microRNAs are involved. Then we will go with which body fluid we will be targeting, either menstrual fluid or saliva or urine, based on the sensitivity of the technique. And then Dr. Professor Suman will be involved in making a device to detect the disease. So this is how the objectives are framed. And then So we got ethical clearance, and we have collected some 18 samples. Actually, this was an ongoing project, so we could take the ethical clearance prior, and this was on microRNA only. So we got 18 endometriotic patient samples, and we collected blood, utopic endometrium, that is the inner lining of the uterus, and the ectopic endometrium, that the endometrium which grows outside the uterus. So we have collected all the 3 samples, and we wanted to first check whether there is any polymorphism or SNPs or mutations at the genetic level from the 3 things. And we are also looking for any somatic mutations. So because the germline mutations may not happen with endometriosis, because endometriosis is not basically a cancer. So we are suspecting some somatic mutations. So we have collected blood, utopic and ectopic samples, and then initially we have we have exclusion and inclusion criteria, so we have used, and then patient consent forms were also taken, and then we got the ethical clearance, and then 18 samples have been collected, and then this is the inclusion criteria: women of reproductive age diagnosed with endometriosis, properly diagnosed with the gold standard, laparoscopically diagnosed. And then exclusion criteria: females with adenomyosis, ovarian cysts, fibroids, ovarian cancer, and polycystic ovarian syndrome are excluded from the study. So we have extracted the DNA from all these samples, the blood and also the tissue samples. This is the representative image. So we could get the pure DNA, and this is 0.8% agarose gel. So we could get from both blood and tissue samples, utopic and ectopic tissue samples, we could isolate the DNA. And then we have initially, so based on the literature, we have identified some 6 miRNA regions with the SNPs. And then we have made the primers. And then from that 6, we have chosen 3, because means we are waiting for more funding. So this, as of now, we have selected 3 SNPs in the miRNA 146A, and there are the 2 SNPs in the miRNA 146A. So this is the RS number, RS5709539 and RS2431697. And another important miRNA let7 family. So we have chosen this particular SNP, RS108778887, and then initial experimental validation, we have taken this, and then we have done the PCR amplification, and we have optimized the PCR conditions, and these are the bands what we have received, which are of this typical molecular, the amplicon size is matching with the size in the ladder. And then so then we have confirmed using Sanger sequencing, we have confirmed the amplified region is miRNA region. And then, so it showed that the primers are designed properly, and then the PCR has been amplified, the actual amplicon of the desired region, and the sequencing results validate the results what we have obtained from the PCR. So we stand here right now. So we need to do more samples. So we need to validate, and then so we need to make use of more literature and also probably AI, so that we can narrow down to few, at least 6 to 8 microRNAs, which play a very important role in the disease. And then further we need to validate. So this is the summary. So what we have worked with, we have completed so we got the ethical approval, of course, prior only. So we collected samples, DNA extraction has been made, and then PCR to amplify the target regions of microRNA genes. And then we have amplified using the PCR, and we have standardized the PCR conditions. And also we have validated that, confirmed the amplicon is of that particular target sequence, what we are looking for. So we stand here as of now, a small baby step. So we should see how it goes. And then we require a lot of best wishes, and then no female should suffer with endometriosis. So we don't know how strong we are until being strong is the only choice we have that holds for endometriosis. Thank you very much. So any questions? I have trimmed the thing a lot. So because other OPI should also get accommodated. I'm done with my thing.
Thank you, Professor Manjula. If there are any questions, please feel free to ask.
Yeah, from
Otherwise,
anything about the disease. Otherwise, I'll stop sharing.
Yeah. Professor Someshwar can continue with the talks.
So, no questions. So I'll stop sharing, and I'll mute myself. Thank you, ma'am. Take care.
Professor Someshwar, you can start. We can't hear you.
Good morning. Thank you for giving this opportunity, Madam, Professor Vijayalata Madam, and Central University Professor. And this is in this project. In this project, I have involved in two tasks. One is task 9 and another one is task 14. In the task 14, in the task 14, first already discussed by Professor Manjula. In that, I involve only in synthesis of nanomaterials for the identification of identification on the lab on the chip by femtosecond laser technique in nanoinvasive method. In that, I have synthesis silver-based nanoparticles and titanium tellurium-based perovskite materials. These materials are made up of with ion exchange method, and then we can move to the core shell with some other metal ions, and we can get on the top of this metal we can antibody, and then we can DSA, we can get this one. But initially we success up to the nanomaterials, the synthesis of this nanomaterials by using parent component, that sodium titanium based material tellurator. We exchange with the copper and silver, and we can get whatever the year in the Raman spectra, whatever we are instrument using for the Raman spectroscopy for identification of this rays, the enhancement of compared to the parent and copper, the silver is 10 times enhancement in the surface, enhancement in the surface of the when we are introducing silver mark. So therefore, this is act as a plasmonic resonance, and we can get the TEM images of this one, and like this, the molecule, the nanoparticles are easy on the surface of the glass. It is well arranged, and we can get the single crystallinity, high crystallinity, and we can get useful for the identification of endometric analysis. So this is the one part. Another one is task 9. It is a synthesis in the task 9. This is a non-invasive identification of glucose molecule in the presence of a device. Whatever the device is based on the OFDV device, they are using synthesis of boronic acid function as organic molecules. These organic molecules consist of 4 stages. First one is the Nogalen condensation. Second one is a nitro group converted into aldehyde. Nitrile group, CN group converted to aldehyde, and third one is aldehyde is react with the boronic ester. We can get Sibase, and then we can this ester is converted into acid by in presence of sodium periodide. We can get the acid based on this microcyclic boronic acids. We already synthesis whatever the obtained compound we are already studied for the fabrication on top contact bottom gate based device, and based on this device results we can obtain that the film is a well organized film, and we can get the measure initial mobility of the initial device is 0.79 centimeters per velocity second. After introduction of saliva on the surface of this device, we can reduce the mobility. So 0.45. So this based on this reduction, the electronic contract and whatever the on-off ratio and everything is changes based on this mobility data. We calculate by using mathematical equations, the delta mu by delta mu naught into equal to S into concentration of glucose. The S value we can obtain from the graph, and this value is 0.11, and the difference between the initial and final we can get the concentration of glucose is 3.91 millimolar. So this is the mechanism actually. What is the basis? This first we can take sample, and we keep the sample on the surface of the device, and interact between the non-glucose and boronic acid by using non-polar interactions, and the current is changes, and we can obtain this is with this mechanism we can get a 3.91 millimolar concentration. At the same time, we are concentration also general blood and also interstitial fluid and sweat also. All four samples we are get optimized results, and this is whatever the reported data is matched. So therefore our devices work well. So with this, we are also more number of practical prototype devices. Also, we need to collaborate with Suman and biomedical department, Professor Suman, and we can get the final. This is about our work, whatever the progress from last 4 months. Thank you for giving this opportunity.
Thank you, Dr. Someshwar. Any questions? Please feel free to ask. If there are no questions, then I'll invite Professor B. Sirisha and Dr. Vijay Bhaskar Reddy to please.
Yeah, thank you, ma'am. Dr. Vijay Bhaskar Reddy will be starting, and then I'll be continuing.
Okay, madam.
Madam, I am unable to share. Yeah. Madam, am I audible?
Yes, sir.
Good morning, ma'am, respected dignitaries and the faculty members. Today we are presenting our research work. The first part of presentation will be presented by me, and the second part will be presented by Professor Shirisha, ma'am. Our work is under task 11, UV visible IR terahertz based time domain spectroscopy and imaging systems for sensing biomarkers under sub-package 2.1. Our work title is Design of Metal Chelates and Ecotype Analysis of Gymnema sylvestre and Withania somnifera for diabetic applications by using UV IR terahertz based time domain spectroscopy and imaging systems for sensing biomarkers. This work combines medicinal plant research, phytochemistry, synthetic chemistry, and advanced spectroscopy to develop innovative therapeutic and diagnostic approaches for cancer and diabetes. We selected two important medicinal plants. One is Gymnema sylvestre, which is a tropical plant, will be collected from southern part of India, which is also known as Goodmer or sugar destroyer, well known for its anti-diabetic properties due to its gymnemic acids. And second plant is Withania somnifera, also called as commonly ashwagandha, which will be collected from northern part of India, northern states of India, which is a subtropical to temperate adapted medicinal plant rich in withanolites, known for anti-cancer, antioxidant, and adaptive genetic activities. These two plants were strategically chosen because they represent different ecological adaptations and phytochemical diversity. Their medicinal properties vary depending on environmental and genetic conditions, leading to the formation of different ecotypes. Therefore, our study focuses on identifying allied ecotypes with superior therapeutic potential using DNA barcoding, phylogenetic analysis, and phytochemical profiling. Another important aspect of our work is the synthesis of metal chelates. In this approach, biologically active phytochemical ligands from these plants are combined with essential metal ions such as zinc, copper, iron, and magnesium to form stable bioactive complexes. These metal chelates can improve bioavailability, stability, and therapeutic efficacy uh, compared to normal phytochemicals. Present work is
Sir,
taken with
slide
the...
change. Yeah.
Yes, I moved to next slide, madam. Can you see it?
Yeah, yes, sir.
Yeah. These are our objectives: to collect ecotypes of Gemina sylvestre and Withania somnifera from different biogeographical zones of India. This Gemina sylvestre will be collected from southern states, and Withania somnifera will be collected from northern states of India. To perform phylogenetic analysis and DNA barcoding for genetic diversity assessment, to conduct a total phytochemical profiling and quantify gymnamic acids using GCMS, LSMS, and HPLC, to design and synthesize the metal chelates of biological active benzothiazole derivatives and key phytoconstituents which are derived from these two plants. To evaluate in vitro anti-cancer and anti-diabetic efficacy of phytochemicals and their chelates, to explore terahertz spectroscopy for rapid marker detection and validation of phytoconstituents. So far, we have collected 3 plants within the state, Telangana state. One is from Khammam and the other one is from Hyderabad, Nirmal. We have collected the plant saplings and we are growing them in our nursery at Osmania University. And if you see the medicinal significance of Gemina sylvestre, it's also called commonly as the sugar destroyer. It's a perennial medicinal plant widely used in traditional Ayurvedic medicine. The leaves of Gemina sylvestre are traditionally used to manage diabetes mellitus by reducing blood glucose levels. It also suppresses sweet taste perception and sugar carvings. It improves the pancreatic function and insulin secretion. It also supports the weight management and lipid metabolism. The medicinal relevance of Gemina sylvestre is largely associated with its bioactive phytochemicals, particularly gymnamic acids. These are the major bioactive constituents, and other phytocomponents are coumarin, flavonoids, alkaloids, tannins, and saponins. Gemina sylvestre has various therapeutic activities, particularly preferably antidiabetic activity. Gymnamic acids reduce the intestinal glucose absorption, stimulate insulin release from pancreatic beta cells, promote regeneration of pancreatic tissues, improves glucose utilization in peripheral tissues. It has antioxidant and anti-inflammatory effects also. It lowers the cholesterol and triglyceride levels and improves the metabolic health. Preliminary in vitro studies suggest that phytochemicals from Gemina sylvestre also work on cancer through apoptosis induction and oxidative stress. And Withania somnifera, another plant which is also called as ashwagandha. It has major bioactive compounds, withanolites. Apart from these withanolites, there are other key components like vithaferin A, alkaloids, saponins, and flavonoids. So our aim is to collect various ecotypes from different parts of India, northern states and southern states, so that we will identify key plants which are qualitatively and quantitatively more viable for this work. So the ecotypes variability is because of environmental factors, genetic factors, and with these it gets the pharmaceutical importance. Environmental factors like temperature and rainfall influence the metabolite synthesis, soil nutrients, and salinity affects the alkalides, flavonoids, and ethanolides. Drought and UV stress can enhance the antioxidant phytochemical production. So geographical locations leads to the variation in gymnamic acid and ethanolide contents. So with this key idea, we have chosen this, and we'll apply various techniques, particularly DNA barcoding technique, to identify our genetic variations. And so far, we have collected these and shade dried and powdered and put it for succinate extraction. And thank you, ma'am, for giving this opportunity. Next part will be presented by Professor Shirisha, ma'am. Over to you, ma'am.
Yeah, thank you, Dr. Vijay Bhaskar. Next slide, please. Yeah, the methodology and all up to maximum extent, Dr. Vijay Bhaskar has completed. The collection of the plant material and all is under the process. Then in the laboratory, we are also not only the collecting and extracting from the oxalate extract we have completed, and then separated the active component by rota evaporation. And we are also planning to synthesize the complexes with the biophyto components. Then the metal chelates are also synthesized in the laboratory. And then we'll be doing the cytotoxic studies on the cell cancer lines. And we planned to work it on 3 cell lines, the HeLa, the cervical cancer cell line, and the MCF7, the breast cancer cell line, and A549, the lung cancer cell line. The next slide, sir. Then the instrumentation, this is the work on the UV visible IR terahertz spectroscopic imaging of the cell lines will be carried out at the Central University in the School of Physics. There, Professor A.K. Chowdhury, sir, is our host from the HCU. So all the samples and all the preparation is done, and the normal basic IR and UV spectrum have been recorded in the various concentrations, and we found that at the 10 power minus 5 to 6 in the micro level, the peaks have been identified very well. And so now the samples are ready to be imaged on the terahertz spectroscopy instrument. Next slide. So here the heterocyclic compounds, as you all know, they are very widely distributed in nature and are particularly important because of the wide variety of physiological activity. Then several of the important compounds containing the heterocyclic rings are in the nature, like the vitamin B complex, alkaloids, antibiotics, chlorophyll, and even the nucleic acids like DNA. Here we have chosen the heterocyclic moiety benzothiazole with a distinct pharmacological profile, including the anti-tumor activity. The biological properties like the antibacterial and anti-inflammatory compounds are shown mostly when this benzothiazole moiety is substituted at the second position. Next slide, sir. So here are the many reported compounds with the various activities at the second substituent position. And the intensive research on this benzothiazole ring system is going on to identify the novel lead compounds to fight against the different cancers, and few recent trends in the field are shown in the figure. The next slide. So in the laboratory, we are synthesizing the benzothiazole carboxyhydrate hydrazide, and then we are then followed by its derivatives, and then synthesizing the metal complexes, and then going ahead with the application part. Then in the first step, the benzothioazole has been prepared by 2-ethyl-2-benzothioazole carboxylate and diethyl oxalate by taking in 1:2 molar ratio and refluxed on water bath with the temperatures ranging from 147 to 93 degrees, reducing by 10 degrees at every 1 hour, and then pouring the reaction mixture in HCl water and ethanol mixture, and on stirring, the ester has separated out, and then it is mixed with the hydrazine hydrate, and then on stirring for 10 minutes, the yellowish compound of the benzothioazole 2-carbohydrazide has been separated. And this will be our intermediate compound. And then we have synthesized N-thiophene 2-carbonyl benzothiazole compound by refluxing with the benzothiazole carboxyhydrazide. Then the compound has been all the spectral recordings have been made, and it is characterized by IR, UV, mass, and NMR spectroscopy. And then apart from this synthesized compound, we have also purchased the phytoligands rutin and then quercetin. This rutin is a flavanol glycoside, and it acts as a potential oxidant and neuroprotective and anti-inflammatory agent found in buckwheat, citrus, and various plants. And this quercetin is a prominent polyphenolic flavonoid antioxidant found in fruits and vegetables. So we have synthesized the complexes of the synthesized benzothiazole carboxyhydrazide derivative, and also with rutin and quercetin. The methyons used for the complexation are cobalt 2, nickel 2, copper 2, and zinc 2 complexes. Next slide, please. Now the terahertz instrument. It is a comprehensive integrated instrument capable of covering the UV, visible, and IR and the terahertz range, and often it is referred to as ultra broadband spectrometer. So we are going to use this instrument for imaging our compounds and the cell lines before the treatment and after the treatment. There are many applications of this technique. It acts as a biomarker and the bioimaging, and it also acts as in the fingerprint region for the biomolecules. And the expected outcome of our research work is expected to produce several impactful outcomes. And we basically classified them into 4 types: scientific outcomes, therapeutic outcomes, technological outcomes, and societal outcomes. So in the scientific outcomes, it's all the entire identification of the elite ecotypes. And this is because of the sophistication of the terahertz instrument, which can give the imaging of the compounds, and we can, by the imaging of the compound, we can also get the mechanism of the action. Then the therapeutic outcomes, like the improved anti-cancer and anti-diabetic agents on the activity of our compounds, and the technological activity with the low-cost point-of-care diagnostic technologies. And then the societal outcome, it would be low-cost equipment and the support for the personalized medicine and contribution towards the translational phytopharmaceutical research. Thank you. We thank the University of Hyderabad and Osmania University for giving us the chance of being the part in the ANRF project. and our host, Professor A.K. Chowdhury, sir, School of Physics, HCU, and Professor Vijulata, ma'am, PI of ANRF project at OU. Thank you, ma'am.
Thank you, madam. So shall we break for lunch and then we'll join back at 2 o'clock?